happily, we're talking about happily. Well, there's also some other terminology that you need to be familiar with. And we kind of hit on it a little bit on Tuesday, but not really. So there's some terms like genes and alleles. So I spoke about that briefly on Tuesday. Can anybody give me an explanation of what those are?
An allele is found on a gene, correct, which are on specific spots of the chromosome.
Very good.
Cool.
Yeah. So if we're looking at a chromosome, okay, then we have a specific area which we refer to as the loci. The loci represents the location of a particular gene. So this little square there represents that little area. So that's my gene. And then the loci represents the location on the chromosome that gene is found. Okay? And so since we have pairs of chromosomes, right, that means that I have another chromosome that is going to be similar, okay? So if somebody tells you same, they are incorrect, okay? Homologous does not mean same, it means similar, okay? Because if it was the same, then that means they would be coding for the same thing, okay? But they should not, I shouldn't say usually, they should not code for the same thing necessarily, because we hopefully made with somebody who is genetically different, right? So they are similar, okay? So if I look at, let's say this is chromosome 1, I'm going to put mom there, chromosome 1 from dad, okay? They are going to be similar, meaning if I'm looking for specific genes, those genes will be found at the same loci on each chromosome, regardless if it's from mom or dad, it's the same location, okay? It'll be the same gene, okay? So I'm looking for gene A. So if I go over here to chromosome 1 from dad, I go to the same loci, I should get gene A, located in the same spot. Okay, everybody's good so far? Now when I look at gene A though, it may be coding for a different expression, okay? So genotype represents my genetic makeup. So basically, what is gene A coding for? That's the genotype. Now, what I express physically is the phenotype. Okay? Everybody's good? So if I'm talking about, we'll just say hair color. Hair color, eye color, skin color, it's actually more than one gene that determines that. And they're located on different chromosomes. Okay? And so when we bring all those genes together, that's why we get this whole rainbow of skin color, variations of eye color, as well as hair color. Because there's multiple genes, and when you have multiple genes, that gives you a lot of different variations, okay? And so we put together those genes from mom A, or from chromosome 1 from mom and dad. And so let's say that mom's gene, we'll just say, skin color, or eye color. Mom says, I'm supposed to have blue eyes. Dad says, I'm supposed to have brown. Okay. So what happens is that we look at these genes, we look at them in the same location, and we look at what they are coding for, and we put them together and say they are alleles. Okay? You say allelic combination, you'll hear that a lot. Okay? Means the combination of both of those genes on those homologous chromosomes. Okay. Now, when we look at alleles, or the allelic combination, we usually have a dominance and a recessive. And so in genetics, if it is dominant, we give it a capital letter. If it's recessive, we give it a lowercase letter. Okay. And so a lot of research has been done on different traits. Okay, and so traits are basically what is expressed, what we see physically. Okay, and so brown hair is dominant, brown or black hair. Oh, I'm talking about eyes. I'm sorry. Brown eyes are dominant, and blue eyes are considered recessive. Okay, and so when we look at the allelic combinations, we either say we have homozygous. Homozygous means the same on each allele. Okay? And we make it specific. So we don't usually just say the individual is homozygous for this trait. We say the individual is homozygous dominant or homozygous recessive. So if it's homozygous dominant, that means that they're containing both of the dominant alleles. Both of the dominant traits on each chromosome. If they are homozygous recessive, that means they have the recessive trait on each. Okay? Everybody's good? Okay. So this individual here has the blue, which would be the lowercase, and it has brown eyes, which is the dominant. Okay? So this individual is heterozygous. Okay. Okay, everybody's good? Yes, no? Okay. All right, so when we're looking at the results of mating, okay, and so this is the idea of segregation, meaning that when we go through meiosis, our offsprings are going to have a combination, okay? We refer to it as segregation, and then incorporated in there is the concept of independent assortment. Independent assortment means that we just mix up all that genetic material and we randomly separate. It's a random separation. So we get, so like males, okay? Males produce like, what is it? 300 million, I don't remember, sperm cells in a period of time. Each of those sperm cells that are produced are different. None of them will be the same. Okay? Because every time we have prophase 1, we mix up that genetic material so that we get 4 unique sperm cells. Okay? So we are always mixing up that material and separating. Okay? So if an individual is in this situation where they're heterozygous, that means in one offspring, or some of the offsprings will get that dominant allele for brown eyes, and the other gametes will get the recessive trait for blue eyes. Okay? But these are just these 2 genes. Remember, we have hundreds of thousands of genes. So that means that every time we have an offspring, the genetic makeup should be different. Okay? Everybody's good so far? So when we look at 2 individuals in there and when they mate, okay, we basically use a process called a Punnett square. Okay? And so Punnett square helps us to basically assort our alleles and see what would be the combinations we would get from these 2 particular individuals. Okay? So when you do a Punnett square, let's say that we are crossing two heterozygous individuals. I'm gonna keep my stuff, my letters this side. Okay, so I'm crossing two individuals. These are considered my parental. Okay, and so when you create a Punnett square, you're going to look at each individual separately. So this particular parent can give me a capital B and a lowercase b, right? So if they produce their gametes, those are the only two possible gametes that can occur. Same with this particular parent here. Okay? So I just match them up, right? So this big B with this big B, right? Capital B, lowercase here. Capital B, lowercase b, lowercase b. Okay? So from this two heterozygous individuals, it has the ability to produce one offspring that is homozygous dominant, two offspring that are heterozygous, and one offspring that is homozygous recessive. This is our genotypic ratio. Okay? It's genotypic because we're looking at the allelic combinations. Okay? But if we look at the phenotypic ratios, we end up with 3 individuals that are going to have brown eyes and only 1 individual that will have blue eyes. Everybody understand how that works? So activity A in your lab is going to be working through some questions. And these questions are giving you scenarios of individuals. And they're looking at tongue rolling. So it has several questions like what would be the genotypes of the F1 generation, which is your first cross. Then it says, okay, so your parental, it changes and it has a different combination of individuals who are mating. And then has several questions about what would their outcome be or what would their offspring look like. Okay? And so it has you do a Punnett squared. And so this is referred to as a monohybrid cross. Okay? Because we're only looking at one trait. So mono means one. We're looking at one trait. So it has you work through several monohybrid crosses. Okay? And so a monohybrid cross, if it's just a dominant recessive, there's no variation, then you would expect to get the 3:1 phenotypic ratio, a 1:1 genotypic ratio. Okay? That's what you would expect to see. Okay? Bless you. So for activity A, that's what you're going to do. And then activity B, you're doing some monohybrid crosses. Okay? Activity C, you are now going into a dihybrid cross. So dihybrid crosses, we are now looking at 2 traits. Okay? And so the crosses that they're doing here in your books, or in your handout here, they're not saying what the particular trait is. Okay? So with the dihybrid cross, we're looking at 2 traits. Okay? So we already have B, okay, and let's add A to the scenario. Okay? So if we're looking at 2 individuals, and so this particular individual is heterozygous for the B alleles and heterozygous for the A. And we cross this individual with another individual who is also heterozygous. Now our chart is going to be much larger. Okay? So what will be the possible alleles or the possible gametes that can be produced from this individual? Remember, they're only going to get one A and one B in each gamete. Anybody know? You want to help me out here?
Homozygous dominant.
So homozygous dominant, right? What else?
Recessive.
Right? Those are the possible gametes we would expect to see from that individual. Because remember, independent assortment means we just mix it all up. Okay? And so we're looking at just those two particular genes. Those are the only combinations we would expect to see. So if this individual goes through meiosis 1 and 2, one of the gametes would possibly have both of the dominant A and B. One gamete would have the dominant A recessive B. One gamete would have the recessive A, the dominant B. And then one gamete would have both of the recessive traits. Okay? And so these are our 4 possible gametes. Okay? So then when we look at this individual, we would expect the same. Oops. Uniform there. So we should get 16 squares. Okay? And so we would fill them in. Okay? I won't do it all because I want you guys to do it. So here we would have dominant A, dominant A, dominant B, dominant B. Dominant A, dominant A, dominant B, recessive B. Dominant A, recessive A, dominant B, dominant B. Dominant A, recessive A, dominant B, recessive B. You guys see how that works? So what we would expect to see down here is a 9-3-3-1 phenotypic ratio. You want me to fill the whole thing out? I'll do a second. Dominant A, dominant A, dominant B, recessive B. Dominant A, recessive B, recessive B. Dominant A, recessive B, dominant B, recessive B. And then dominant A, recessive A, recessive B, recessive B. Okay. And so it'll give you a 9-3-3-1 phenotypic ratio. Okay. Everybody's good so far? And so basically we'll have 9 individuals that will have the dominant trait. So like this individual is dominant for both. This individual would be considered dominant for both because it has that dominant B. This individual would be considered dominant. They're all going to be up here. All right, that's 1, 2, 3, 4, 5, 6. Right? And so 9 of them will be dominant for both traits. 3 of them will be dominant for one of the traits and recessive for the other. 3 of them will be dominant for one of the traits and recessive for the opposite one, right? And then only one is going to contain all recessive, which is this one here. I'll go ahead and fill it out so we can make sure y'all see this. And I'll use different colors. So the black dots represent those that were dominant for both traits, right? So if we count them up, that should be 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 are going to be dominant for both traits. Let's do the ones that are dominant for A, recessive for B. So it's 1. There's 2, and there's 3. Okay. And so then, those that are going to be recessive for A, dominant for B is 1, that's 2, and there's 3. Okay? And then my 1 that is going to be all recessive is right here. Okay, see how that works? Okay. So that is what Mendel's expected ratios would look like. A 9-3-3-1 phenotypic ratio. Make sure you understand if it's phenotypic or genotypic. Most of the time we focus on the phenotypic ratio. Okay? So the second activity, or what is it, activity D, I believe, as in dog, goes into predicting the outcomes for dihybrid crosses. So it gives you several scenarios that ask you then to write out the possible gametes, do a Punnett square, and then what are the list the possible genotypes of the offsprings, and then list the possible phenotypes of those offsprings. At the back of this exercise, it has some questions. Okay. I would like for you to do question 1 and question 2 so that I can check it and we can talk about it, make sure you know how to do it. Okay? And then the other ones use as practice. Okay? Questions for me?
Questions 1 and 2, you said?
Yes.
On page 17?
Yes. Are there any questions? Yes, ma'am.
On that second Punnett square, did you use only black and orange?
Oh, I
I
should use
can't
it.
tell the difference.
Yes, let me
I'm
use
sorry.
blue. I'm sorry. No, I'm sorry. Let me use blue. Is that better? So use black, red, and then blue and green.
So which ones are the red ones? What are those indicating?
So these are the dominant A's, recessive B.
Okay.
And so that's one of your 3s. And then the blue ones are the ones that were recessive for A, dominant for B.
Okay.
And then the green is the loner. It's all recessive.
Thank you.
You're welcome. So you're going to work with a partner or in a group, however you would prefer. Okay, but I want you to work with somebody. I don't want you to just do it on your own. Okay, I want some discussion along the way.
We just started. Oh, you wrote it down. Do you mind if I?
Oh yeah.
Yeah, I was trying to figure out.
I just know that I can roll mine and my dad can't, so I'm just be like big or little R.
I think I'm pretty much the only one in my family who can't. So
You can't write your time?
So your parents are both out of Vegas.
my sister can. My brother can. I can't.
And then I'm pretty sure it's just asking.
I haven't even.
It says after meiosis, each gamete has one number. So I'm thinking this is just like one of each. So
One member of a pair, so that's one, yeah. Each person's genotype.
What page are we on?